Okay, chapter 3, nucleic acid structure and function. So in this chapter, we will go through the structure of DNA and RNA, which will be the fundamental knowledge of the nucleic acid. And the nucleic acid is the monomer for the nucleic acid, the polymer, the whole DNA molecule, RNA molecule. And also we will discuss genomics and the methods in nucleic acid biochemistry. Okay, so this is just like a review that you know the new DNA structure and RNA structure, they pretty much are similar. Okay, except these in this sugar number 2 carbon, whether it's hydroxy group or hydrogen group. So that gives you a difference between the DNA and RNA. Otherwise, they all have these three basic component. One is the nitrogenous base, sugar, and phosphate. All right, and then for the nucleoside would be the sugar and the new nitrogenous base that give you a nucleoside, and then you add a phosphate group that become the nucleotide. Okay, and these three components basically they go through this chemical reaction. So for example, the nitrogenous base and the sugar, they have this dehydration and form the glycosidic bond. And here, the number 5 carbon and phosphoric acid, also dehydration, and then form this mono, phosphor monoester bond. Okay, that's how they form this nucleotide. And this is the formation of the ADP and ATP by the additional phosphate group. So from here, you see they form this nucleotide. This is the deoxide because it's number 2 carbon here. It's only a hydrogen ion, it's deoxide. So deoxide AMP, adenosine monophosphate. And then if you add another phosphate group, has to go through this dehydration. See, they remove one molecule of water, so they form this alpha beta. This will be the adenosine 5' diphosphate, and then here will be triphosphate. You just continue to add the phosphoric acid, okay, the phosphate. So that in the end, this is the adenosine 5' triphosphate. And then the nitrogenous base, basically we have the purine and pyrimidine, right? The purine, because the purine here, you have taurine, adenine, guanine, they both are the purine. Pyrimidine, we have cytosine, thymine, and uracil. And they only have one ring, okay? So that's a two different type. We have the pyrimidine and purine, depending on their structure similarity. Okay, and so basically here's the nomenclature for the nucleoside and the nucleotide. So for the DNA here, adenine, so deoxyribonucleic adenosine, right? The full name is deoxyribonucleic adenosine 5' monophosphate. So that's DAMP. And then for RNA, that'll be the adenosine 5' monophosphate. So that'll be AMP, right? So that's the nomenclature. So the structure of DNA, primary structure, basically is a DNA sequence, right? And that can be found in all biological molecules. And then they have the unique arrangement of deoxyribonucleic or ribonucleotide arranged in single chain, and then usually depicted as a single letter in a row, like the A, G, C, T. And then the secondary structure, that's two complementary strands of DNA, the double helix DNA. They've been held together because they have complementary base pairing in anti-parallel fashion. So that's the double-stranded DNA, the secondary structure. And then the tertiary structure, that's a supercoil that we will discuss later. And also this is fourth degree of structure, the interaction between the DNA and the proteins. Okay, so these are the different level of the DNA structure. So here is the secondary structure for the double helix DNA. All right, double helix DNA, and then as you can see that normally the majority of the DNA is right-handed. So right-handed, see from here on the right side, move to the left, right, on the top here, that's right-handed like this, right-handed in the front here. And one turn is about a 10.5 base pair. Each pair is a 3.4 Armstrong. Okay, and the DNA must be condensed to fit within the cell. That's what the that's we know that in the bacteria, right, all right, that fit into this cell, and the eukaryotic chromosome fit into the nucleus, right? And this is because the DNA is associated with some protein, and this protein can help to condense the DNA so that the DNA will be able to fit into the cell. Okay, and this is just to demonstrate that the smallest free living non-symbiotic bacteria, right? The candidates, and the genome is like a zero point 0.7 megabase, and then has 800 gene, and the 0.7 megabase. And remember if you go back previous slide here, so each base pair is about 3.4 Armstrong, which is 0.34 nanometer. Okay, so, so it's a 0.34, and then 0.34, and then if you multiply nanometer, multiply 0.7 mega base pair. Okay, so if I do the calculation, this is 0.7 mega base pair, so now 700 kilobase, all right, 700 kilobase pair, and times 0.34 nanometer, so that's a 238 nanometer. So 238 nanometer, that's like about a millimeter. So it's 238 millimeter, 238 millimeter, and that would be 23 centimeter. And normally the bacteria cell is like 100 nanometer. So that tells you that you have to condense so much, at least 1,000 to 3,004 to fit into the cell. Okay, so that's the calculation, so that you know that the DNA must be condensed so that they can fit into the cell. And then the complementary base pairing. So we know that the guanine cytosine, right? Guanine cytosine. See my point here. So the guanine cytosine, the 3 hydrogen bonds. See the hydrogen, oxygen, hydrogen, nitrogen, hydrogen, oxygen. Right? 3 hydrogen bond, and adenine thymine, the 2 hydrogen bond. So they form this base pairing. That's complementary. Okay, and that's based on this Chargaff rule. So basically, two long polynucleotide chain are coiled around a central axis. They form a right-handed double helix. Okay, and the two strands are anti-parallel. It's 5-3 orientation. They run in opposite direction. And each complete turn helix is 34 Armstrong, which is 3.4 nanometer long. Okay, and double helix has a diameter of 20 Armstrong, which is 2 nanometer. All right, 2 nanometer. And the amount of the adenine is proportional to the amount of thymine because of the base pairing. All right, so if you have 10% of thymine, then you must have a 10% of adenine. Okay, has to be the same because they are uh, complementary to each other. And then guanine residue is proportional to the amount of cytosine. And then the sum of the purine will always equal to a sum of the pyrimidine. And then the percentage of G plus C is not necessarily equal to the percentage of A plus T, because let's say if we in the genome you only have 10% of A, that means you only have another 10% of T, so that total is 20%. And that means the other G plus C will be 80%. So G plus C 80%, that would translate into the 40% is G, 40% is C, and then 10% A, 10% T. So that's why the percentage of G plus C is not necessarily equal the percentage of A plus T. Okay. And base staking, right? So base staking is just like a base staking. The neighboring base pair, right? They have to shift 36 degrees because one turn is 360 degrees for 10 around 10 base pair. Therefore, each pair, neighboring base pair, they shift 36 degrees. Okay, and also the base pair, the base has to orient it so that the hydrogen bonding with another base required they are arranged in planar fashion, parallel to the adjacent base on the same strand and located in the interior of the helix. And the base pair are stacked upon each other within the van der Waals distance. Okay, so that means from this, the top to the bottom, right? They stack upon each other within the van der Waals distance, so that they have the stability so they can provide the stability through the hydrophobic effect and the van der Waals interaction. Okay, and the here just to show you that they are commonly seen three different type of DNA structure, but the majority of the DNA structure is the bDNA structure. It's about 90% to 92% in the nature of the B form DNA, like this one, B form DNA, right-handed, right hand from right to the left, right in the front here, right-handed, and A form DNA also right-handed. Is a short and wider than the B form DNA, and this A form DNA only occur when it's a highly dehydrated condition. Also, sometimes we also can find a Z form DNA. It is left-handed, so from left to right in the front as left-handed DNA. That's most narrowed DNA form. And so the double helix DNA, basically they form this double helix and through those complementary base pairing. That's the hydrogen bond, the 3 hydrogen bond between G and C, 2 hydrogen bond between A and T, and this hydrogen bond can be broken so that the double-stranded DNA become a single-stranded DNA. So this process to break the double-stranded DNA into the single-stranded DNA is called denaturation, right? Denaturation, which is also called melting. So that's a that would be the process from the double-stranded DNA to the single-stranded DNA. They separate into the two individual stranded. Okay, and of course the DNA can also renature if you are you if it's in the right condition. Okay, so normally when the DNA will be denatured when we increase the temperature to a certain degree here. Okay, when the temperature is increased, when it's too high, so that this curve x-axis, that's the temperature, degrees Celsius, and then the y-axis, that's a percent single-stranded DNA. So as you can see, when you increase the temperature, the amount of single-stranded DNA increase, all right? And then the amount of double-stranded DNA reduced. okay, until at the point where you have the 50% in the population are the single-stranded DNA, and then the other 50% are the double-stranded DNA. At that point, the temperature is called Tm, right? Tm here, Tm. That's a melting temperature. That's the temperature where the in the population that you will see 50% of the population are the single-stranded, and the other 50% are double-stranded. At that point, we call it melting temperature. And then if you continue to increase the temperature, in the end, it's 100% will be a single-stranded. However, when you decrease the temperature, then the amount double-stranded DNA will increase until about 60 degrees. So almost all the DNA will return to the double-stranded DNA status. Okay. And the hyperchromic shift, there's a difference in absorbance between the double-stranded and single-stranded DNA after the denaturation. That's called hyperchromic shift. And then you can see the different nucleotide. They have different hyperchromic shift. So you see the AMP, GMP, this is CMP and DMP. And then, as I mentioned, that if you increase the temperature, single-stranded DNA increase in the population, all right? And also depend on the amount of GC percentage in the sequence. So if you increase the GC content in the DNA molecule, then the melting temperature will increase, because the more GC, then you have more stacking interaction to stabilize the double-stranded DNA, okay? And also another force would be the complementary sequence, right, to help you to stabilize the structure. And so that's why the higher percentage of GC, then the melting temperature will increase. It'll be higher than the low percentage of GC DNA molecule. And then also if the solution's sodium concentration increase, then the melting temperature also increase. Okay, so you can see the three curve here. 50% has the lowest melting temperature, and then 100 millimolar has the highest melting temperature. This is because the sodium ion is a positive, and then the DNA is negative. So the sodium ion can neutralize the negative charge on the phosphate group to stabilize the helix. Okay, so that's why if you incubate the DNA in higher concentration sodium chloride, then the melting temperature also increase. And the DNA supercoiling. So basically, DNA supercoiling, that's a structure where the majority of the DNA molecule inside a cell fall upon that cell. That's a supercoiling. And they can be found everywhere, supercoiling, okay? And supercoiling, all right, so basically, you can see the left in the front, okay, left in the front, this is a positive supercoil, and the right in the front, that's a negative supercoil, okay? See left in the front here, that's left-handed. Left-handed is positive supercoil, and right-handed here, right-handed, that's a negative supercoil, okay? And the DNA topology, so basically, this is important because all the nucleic acid-based cellular activity would be relevant to the DNA topology, because during, for example, DNA replication, transcription, and you will need to open up the double helix. When you open up double helix, you increase the supercoil to the DNA molecule. That's why we also discussed the linking number. So the linking number will be the number of times a strand of DNA wind in the right-handed direction around the helix in the imaginary plane. So LK linking number is the sum of the twisting number plus the Riley number. So the twisting number that's measure the winding or DNA strand around each other, and Riley number measure the crossing of the DNA strand. Okay. And the here is just to demonstrate that so your rotation. So for example, this is a plasmid DNA, circular DNA. Your rotation of DNA result in changes in the twist or wreath, right? The twist that decrease, the rise stay the same. Twist stay the same and rise, decrease. Okay. And now this is right-handed, so it's a negative supercoil, right? So you see that when you rotate the DNA, then you introduce this negative supercoil, and then if you are, this is counterclock, and then negative supercoil plus positive clock, the clockwise, right? The clockwise turn that give you the positive supercoil. Okay, the twist stay the same, the writing number increase, right? So here's just an example. Okay, so let's say we have relaxed circular DNA with 50 helix turn, 525 base pair, and then B-form DNA, every turn is 10.5 base pair per turn, so 525 divided by 10.5, so that gives you 50. So that means the twisting is 50. And since the linking number equal to twisting plus writing number, and there's no writing number because there's no supercoil here, so the linking number equal to 50 plus 0, 50, right? If you break the phosphodiester bond in one DNA strand and remove the four turns, if you remove the 4 turns, that means the twisting number decrease 4, right? That open up this double helix, so that'll be 4 turns less than the previous one, so that'll be 46. And 50 minus 4 is 46, linking number will be 46. Okay. If you let the DNA, okay, to reach conformational equilibrium, so they form this right-handed supercoil that introduce the negative writing number. So if you have 4 turns here, then that's minus 4, right? And then if you have twisting number 50, so the linking number will be 46, which will be 40 minus 4. 50 minus 4, that's 46. Okay. Nucleosome assembly. So basically, one of the cellular activity that can introduce the writing number, the supercoil will be the histone. So here, just to show the relaxed DNA, if you assemble the nucleosome, right? So the histone core attached to the DNA that will introduce this here, that will be negative supercoil. And then on the other side is positive supercoil. And then we will have the enzyme that can help to cut this DNA, break the DNA, and remove the positive supercoil. Okay, down here. Okay, and then your enzyme-mediated strand break to remove this positive supercoil, then you open up. If you remove this nucleosome, then you have the naturally occur the negative supercoil. Okay, And that's how the histone core incorporate into the DNA to package the DNA. Okay, and then here is, for example, during the for example, the replication, you open up double helix, then you can introduce the positive supercode in front of the replication, or even you open up for transcription, then there will be the same open up, introduce the positive supercode. Okay, and here is a very important video that I want you to look at. Okay, so there you have some idea about the
It is a well-known
supercode.
fact that double-stranded DNA forms a double helix structure, but the double helix itself can twist and turn into different conformations. So, how does DNA change its shape, and why is this behavior important to our cellular functions? This video will use a physical model of DNA to help explain the significance of DNA topology and simulate how specific enzymes modify the conformation of double-stranded DNA. The unstrained or relaxed DNA structure has 10.4 base pairs per turn of the helix. One way to describe the structure of a DNA double helix is by a combination of its twist and writhe, the sum of which is called the linking number. The twist is described as the number of helical turns of one strand about the other, and can be measured by counting how many times one strand wraps completely around the other. For example, this relaxed linear DNA has a twist of 1, 2, 3. There is 0 writhe in this example. The writhe is described as how many times the double helix crosses over itself, and is positive or negative depending on the orientation. For example, the circular DNA has a writhe of -1 because it follows the right-hand rule and the upper strand travels left to right, and this DNA has a writhe of 1. Both of these examples.
So this is the positive because the left-handy, okay, left to right, okay.
Also have twist. Realistically, long DNA molecules like these can have linking numbers in the thousands. For this video, we're going to look at a physical model of circular DNA with covalently linked ends. Unlike linear DNA, this shape is topologically constrained, meaning you can't change the linking number of the DNA without breaking one or both of the strands. But what would physically happen to this DNA if you added or removed twists or writhe in the double helix? Overwound DNA has fewer than 10.4 base pairs per turn of the helix. Thus, overwound DNA is more tightly wound than relaxed DNA and has a greater number of twists and a higher linking number than relaxed DNA. When DNA is overwound and has an increased linking number, the DNA becomes positively supercoiled, and the double-stranded structure begins wrapping around itself and creating positive writhe. Regardless of these visible changes in conformation, remember that positively supercoiled DNA will have a higher linking number than relaxed DNA, and the two strands of the double-stranded DNA are more difficult to separate. Underwound DNA has greater than 10.4 base pairs per turn of the helix. Thus, underwound DNA is less tightly wound than relaxed DNA and has a fewer number of twists and a lower linking number than relaxed DNA. When DNA is underwound and has a decreased linking number, the DNA becomes negatively supercoiled, and the double-stranded structure begins wrapping around itself in the opposite direction as positive supercoiling, creating negative writhe. Regardless of these visible changes in conformation, remember that negatively supercoiled DNA will have a lower linking number than relaxed DNA, and the two strands of the double-stranded DNA can be separated more easily. But if DNA is least strained in the relaxed form, how does the double helix change from being relaxed to overwound or underwound? And how does positively or negatively supercoiled DNA return to a relaxed state? Topoisomerases are a class of enzyme that usually restores DNA to its relaxed state. This means that topoisomerases unwind overwound DNA strands, reducing the linking number back to the preferred 10.4 base pairs per turn, or rewind underwound DNA strands, increasing the linking number back to 10.4 base pairs per turn. Topoisomerases work by breaking one or two DNA strands and passing the same number of DNA strands through the break. This change results in an increase or decrease in the linking number of our topologically constrained circular DNA. There are two main categories of topoisomerase, type 1 and type 2. Type 1 topoisomerase breaks one of the two DNA strands and passes the other strand through the gap. This increases or decreases the linking number by 1. Here, the top DNA double helix represents the structure before the topoisomerase added a twist, and the bottom DNA double helix is after the enzyme completes the reaction. Type 1 topoisomerase reactions do not require additional energy. Type 2 topoisomerase breaks both of the two DNA strands and passes the entire double helix through the gap. This increases or decreases the linking number by 2. Here, the top DNA double helix represents the structure before the topoisomerase acts, and the bottom DNA double helix is after the enzyme completes the reaction. In this case, the result is that the rise of the molecule increases by 2. To make this idea clearer, this diagram shows what this change would look like if you added one positive rise at a time instead of using type 2 topoisomerase. Type 2 topoisomerase reactions require some form of energy, such as ATP or NADH. Bacteria also have a special form of type 2 topoisomerases called gyrase. Gyrase enzymes use energy to negatively supercoil DNA rather than restoring DNA to the relaxed state. Certain thermophiles also have topoisomerases that use energy to positively supercoil DNA rather than.
Okay, so that's the topoisomerase that help to remove the remove these positive supercoil. So later on when we discuss the replication transcription, then you will see how the process introduce the positive supercoil, and then we need these topoisomerase to remove the positive supercoil. Otherwise, the DNA would be under the very high strain. Okay. So she mentioned there are two types of topoisomerase. Okay, type 1 that cleave one strand and reduce the supercoil region by 1 turn. Type 2 cleave 2 strands of DNA, reduce the supercoil by 2 turns, and type 2 require the energy. Okay. So type 1, for example, you have a single strand nick, and then you move this single strand to the other side. Look, so that you can open up. Right, so one strand pass through the other. So this one pass through to the other, or pass to here, so that you can have these and rejoin. Okay, and type 2, the topoisomerase 2, basically topo 2, you see the supercoil. So originally is right-handed, and then you relax and then continent. Okay, or relax, become like this. Okay, that's the topo 2. Okay, so that's the topoisomerase. I'm sorry, these are not the for the chapter 1, so I don't know why these are here. These will be the chapter 2. Right, then the second topic for the genomic, for the chapter is the genomic, and the genomic the genome basically is the collection of a gene, and then the number of a gene increase. The number of gene increase, the more the complex the organism is. So you can see the number of gene increase when the organism is more complex. Okay, and the condensation of eukaryotic DNA. So basically from 2 nanometer of the DNA, 20 Armstrong the DNA, and then associated with the histone protein that's 10 nanometer that give you a nucleosome, and then the nucleosome they pack into they form this higher order chromatin structure 30 nanometer. Then from 30 nanometer they can become the loop chromatin, and then they condense the coil of chromatin to 700 nanometer. Finally, you see that package into a 1400 nanometer chromosome. Okay. And then, of course, those are the condensation from the DNA nucleosome to chromatin. They require the presence of the histone protein. That's the first level of the condensation from 2 nanometer to the 10 nanometer. Require this histone protein. Okay. And the histone protein, we have the cohistone H2A, H2B, H3, and H4 that give you this octamer, the histone core, so that it can wrap around the DNA to package the DNA. And then in the presence of a linker histone H1, then they can even package this nucleosome further into this 30 nanometer chromatin fiber. Okay. And then for chromosome, like you can see that we have a centromere, telomere, and the whole thing we call chromosome. We'll discuss later. And the gene, so basically a gene consists of the upstream regulatory DNA sequence and downstream transcribe region, right? So we have the upstream, that's a promoter, right? Upstream is promoter, and then we have transcribe region of gene. And here we call coding region, and then here is a promoter region. Require the binding of these trans element, the RNA polymerase, transcription factor, okay? And this is prokaryotic gene structure. So we have DNA promoter, right? And then coding sequence. It can be the monocistrone, but for the bacteria, for the prokaryotic cell, mostly they organize into the polycistrone because they can be organized into polycistrone to be controlled under one promoter, okay? And then the eukaryotic gene structure, so you have the exon, intron, and then only the exon can be joined together to convert it into to be translated into the protein, and the intron will be removed. Okay. And because these exon, they can have a different combination, so the exon shuffling that can give you a different genetic diversity, okay, because different exon combined can produce different protein. Okay, the bioinformatics basically is the field used in biochemistry to discover the function of unknown gene, like the human disease gene that can be targeted with the pharmaceutical drug. That's bioinformatics. So it can have the NCBI blast search are used to analyze the information. That's bioinformatics. And this is just the example, to identify this ICMT mutation in the Hutchinson-Gilford the progeria, because from the bioinformatics they identify the particular nucleotide, and there's a single nucleotide polymorphism. So one nucleotide change, so that would make change into the protein, and then cause the mutation and cause the disease. Okay. So that's a single nucleotide polymorphism. So that's single nucleotide mutation, right? So you can have, for example, in this position can be A or can be T, and that's the polymorphism. Or it can be even the deletion, single nucleotide deletion or insertion. That can always give you the mutation, okay? And short tandem repeat can have deletion, insertion in the genome, and the variable number tandem repeat. That means they have a different number of repeat in the genome. Okay, that all give you the polymorphism. And here just to show the polymorphism used in DNA analysis, so that, for example, in this case, this maternal paternal. So we remember we have two chromosomes. One from the mother is maternal, from father paternal. So they say in the victim here, then the VNTR1 and VNTR2, VNTR3, right? So we have these different lengths of DNA, And then after the PCR and then electrophoresis, you see the pattern is like this. And now if you have the chrysin sample with these different pattern, and you can observe these. Let's say, for example, the VNTR1, the green one. So you have short one, long one, and then chrysin is long and long, right? And the victim is long and short, and these both are long, and the other one is even longer. And then suspect A has a short and long, right? Short and long. And then suspect B has this long and long. So these two match these chrysin sample. And also if you look at VMTR2, the suspect B also match this pink one, and then brown one. So this is the application of polymorphism in the forensic science. Okay. And then precision medicine basically is a method used to tailor the treatment of a disease to fit the genetic material of the individual. That's a precision medicine. Okay. And normally for the precision medicine, you need the help from the DNA sequencing. Especially now we have the NGS, next generation sequencing, that can help to determine the person's whole genome, and based on the person's whole genome, then you can have specific design of the need for the drug, and that's a precision medicine as well. And for the nucleic acid biochemistry, so the first thing you need to know is the plasmid, which is the self-replicating piece of circular DNA that can carry the extra genetic information not contained in the chromosome or DNA, and can be cloned, conjugated, transformed, or transduced. Okay. This is to show you the how to use the plasmid to transfer the genetic material. Can be the transformation. Transformation basically you incubate the bacteria cell with the DNA, and then the DNA enters the bacteria cell. That process is called transformation. And conjugation, basically you have two cells come to close proximity, and then they form a tube, and then the plasmid DNA can be replicated and transferred to the other bacteria cell, and that process is called conjugation. And transduction basically is it transferred by the bacteriophage, because the bacteriophage infect the bacteria 1 and then carry the DNA from the bacteria 1 and then bring it to the bacteria 2. And this process is called a transduction. Okay. And plasmid is important because they can be used as a cloning vector. So the cloning vector to carry a foreign DNA sequence so that you can study or express the gene. Okay. And for the plasmid to act as the cloning vector, it need to have this ampicillin resistant gene, which can be used as a selectable marker to identify the recombinant cells. And also it need to have original replication, so they can replicate the DNA, the plasmid DNA before the cell division. Okay, and also the plasmid need to have the multiple cloning site, it's MCS. That's the place where you can clone the foreign DNA into the vector. Distribution enzyme is commonly used enzyme in the biotechnology, because the endonuclease, they can cut the DNA, the type 2 endonuclease, we call it a restriction enzyme. They can recognize the specific DNA sequence and cut the DNA at that recognition site. That's a type 2 recognition, type 2 restriction enzyme. And so that's why it's important to use it for the biotechnology. So this is just an example of type 2 restriction enzyme. So for example, they have this recognition sequence, GAATTC, and the palindromic that reverse the sequence also GAATTC. And then they recognize this sequence and then they can cut it here. All right, so after you cut it, then you dissociate these two fragments and then produce a 5' overhang here. And then for this PST2, PST1, they recognize CT, GCAG, and then they cut between A and G here. Right, then you have two fragments, okay? And this is 3' overhang. And then this is blunt end. So basically small one, recognize GCC, GGG, and CCC, GGG. And then you can cut in the middle as a blunt end. Okay, that's the type 2 restriction enzyme can produce 3 different types of the overhang. Can be 5' overhang, 3' overhang, or just blunt end. So the recombinant DNA technology, basically you have the vector, right? You take the vector and then you have a multiple cloning site and you cut this multiple cloning site and put a piece of DNA into this cloning site. And then in this process, we also break this LEGZ gene, the LEGZ gene, so that they would no longer produce the LEGZ beta galactase, so that in the presence of X scale, the colony will be white. All right. If this gene is not inserted into here, so that the LEGZ will be produced. When the LEGZ is produced, they can cleave the S scale so that you will see the blue colony. And this is also called a blue-white test to determine the recombinant cell and recombinant DNA. cDNA library basically is complementary DNA, okay, generated from the messenger RNA. So as you can see, we can collect population of messenger RNA because they have poly-A tail, okay, and then you use the oligo DT primer, right, bind to the poly-A, and then they start the transcription, okay. Okay, cDNA library here, so collect the population messenger RNA, and then use these messenger RNA as a template, all right, to synthesize the DNA. So you obtain this first strand of cDNA, which is complementary DNA, all right? And then you have ribonucleic edge to degrade this messenger RNA and then form the second strand of cDNA and give you the double strand DNA. And then you can use it to a clone. Okay, high throughput DNA sequencing. Basically, the original method is called Sanger deoxy sequencing. So basically, they use the idea that if the nucleotide with the deoxy 3' end, all right, it's like from originally it should be hydroxy. If you change that to the hydrogen, then the DNA synthesis will not occur. Therefore, the DNA synthesis will stop. So that for this experiment, you just set up 4 different reactions. The first reaction, you mix the dideoxy A with the deoxy ATCG, and then you perform the DNA synthesis, and they will produce different lengths of the fragment. And then you perform the electrophoresis to separate those DNA fragment from the length of a fragment. You'll be able to determine or estimate or predict the exact nucleotide in that exact location. Okay, so that's the Sanger sequencing. So for example, in here, so you mix the dideoxy ATP with this deoxy NTP, and then they say, for example, they produce these 2 different strands, 2 different lengths fragment and run the gel. So you see this, the length of DNA molecule. Then from this location, you can determine what's the nucleotide in that particular site. And PCR reaction, basically it's an in vitro DNA synthesis reaction, okay? And also it can be the length of PCR product can be determined by the design of the primer. And then for the PCR reaction, the 3 temperature phases, so it can be the DNA denaturation, annealing, primer extension, and DNA synthesis. So DNA denaturation normally is above 90 degrees to denature the double helix template DNA. And then you have the primer adhere to the template. Once the primer adhere to the template, then you can do the primer extension and DNA analysis. Okay, just like this schematic diagram. So we have this template DNA. From the beginning, the cycle 1, the double helix DNA, and then denaturation, so denature, here denaturation, and then the primer bind to the template, and then they perform the synthesis. So from 1 copy to 2 copy in the end of cycle 1, and 2 copy to 4 copy after the cycle 2. Okay. So the PCR reaction is 3-step. Denaturation is 95 degree. Annealing basically depends on the melting temperature. It normally is around 60 degree, and then 72 degree DNA synthesis. Transcription analysis. So you can use the microarray, all right, microarray to determine the amount of the transcript, or you can use the NGS, next generation sequencing, using the RNA-seq to sequence all the RNA. That is more accurate to determine the transcription time. So this is just an example to show the gene chip microarray to study the transcriptome, right? And then basically you have this all oligo primer DNA on the slide, and then you add the cDNA, so you collect the messenger RNA from the single-stranded the complementary DNA, and then degrade messenger RNA, make the double-stranded DNA, cDNA, and then you label these DNA, the double-stranded DNA, cDNA, and then mix with these microarray that each place has the particular primer sequence. The probe sequence can help you to identify the source of the transcript, and then based on the amount of the intensity of the signal that show from each position, then the computer can help us to determine the amount of transcript, okay, for that particular gene. So here I show you that the treated cell, on treated cell, you collect the messenger RNA. And from the intensity of the fluorescence, then you'll be able to determine the amount of transcript for a particular sequence. The next generation sequencing, basically there are several different types of next generation sequencing, but they are based on the single sequencing. And there are 4 different ones: the massively parallel sequencing, sequencing by synthesis, sequencing by ligation, sequencing by semiconductor technology. Okay, here is the example to show the massively parallel sequencing. So the parallel sequencing based on the idea that during the synthesis, right? During the synthesis, you will release the pyrophosphate, okay? So only keep the alpha phosphate group, and then remove the pyrophosphate, the beta and the gamma phosphate. And these pyrophosphate can be used to form the ATP, right? Convert APS to ATP, and then in the presence of the luciferase, they can produce the energy, and then the energy supply the enzyme to give the light. So every time when you add one nucleotide, you will emit the light, so that you will know that the nucleic acid, one nucleotide has been added. Okay, that's a pyro sequencing. And the other one is this sequencing by synthesis. It's a reversible sequencing, because each nucleotide they use for the sequencing has the fluorescence dye and also 3' the blocking group. So every time when you add one particular nucleotide, that nucleotide will give you the fluorescence signal, and also it block the continuous DNA synthesis. So you can determine the synthesis one by one, okay? And then CRISPR-Cas9, that's the technique that nowadays scientists use to target a specific DNA region and then to delete the DNA region, because the design they rely on these particular 20 nucleotide that you design, so that can target this whole thing into a particular DNA target, right? Based on the design, the CRISPR RNA, and also the Cas proteins, because this sequence will lead the whole complex to the target, and then the protein, Cas9 protein, will then cut the DNA. Okay, that's called CRISPR-Cas9. So like after they bind to the target sequence, they can cut the DNA. So knock out a gene, for example, or you can knock in the gene. All right, so that's the end of the chapter 3. If you have any question,